I thought what I might do today is talk to you a little bit about the future rather than the present or the past, and share with you some thoughts on where pneumococcal immunization might be headed in the future. So I'll describe a little bit the epidemiology today, and certainly leave at the end time for questions if anybody wants to ask specific questions about pneumococcal disease or immunization or things that are more practical and current. But I thought this might be, since it's an area of very active research, not just in my laboratory, but really throughout the world, I thought this might interest you as a sort of view of what might be coming down the pike. So I have some potential conflicts of interest. I provide advice to Merck on adult immunization programs, in particular their pneumococcal vaccine. I also work with several scientific advisory boards listed here. And then of relevance to this talk in particular, following work that I'm going to show you today, Children's Hospital spun off a company called Affinities, which was funded initially by the Gates Foundation for the development of vaccines for the developing world, an area that has been a strong interest of mine from the beginning. And this company called Affinities is now in the process of trying to develop inexpensive or less expensive vaccines for the developing world, and I'm going to show you one of these examples towards the end of the talk. So in a group like this one, trying to convince you that vaccines are by far, with the exception maybe of water, cleaning water supply, one of the most important interventions that mankind has ever invented, is not a hard task. So I won't really go into much detail on that. I think we are all aware of the tremendous impact that vaccines have had on global health. What is more challenging is to go over why it's so difficult to develop new vaccines today. And one of the reasons is that we really don't understand, in fact, how most of the vaccines that we make work. The older vaccines, whether you talk about pertussis or diphtheria or measles and mumps and all these others that have been accepted and approved for years, when you actually sit down with scientists who study those organisms and say, how does this vaccine actually work, you sort of get a little bit of the mumbo jumbo that we try to avoid talking to with our patients, but is a definite reality. The other issue is that the best vaccines we have, the ones that we all give and we know that, okay, this child is not going to get this disease because this vaccine works so well, represent what we call the low-lying fruit of immunology. These are diseases where if you get the disease once, you will never get it again. And those are pathogens for which vaccines have been very easily developed. The example for it would be mumps, measles, varicella. Obviously, shingles is a complication, but somebody who gets varicella once will not get varicella a second time. Somebody who gets measles once will not get measles a second time. And those are the initial, when you think of what were the first vaccines ever invented, they targeted those pathogens because immunity after natural exposure conferred lifelong protection. Another great example, I think, is the yellow fever vaccine that you heard my friend and colleague Christy Moffitt talk to you about today. Yellow fever vaccine is a phenomenal vaccine, and the disease itself, while potentially catastrophic, leads to lifelong immunity. So whether you get the disease, which I hope not, or the vaccine, you will be protected against yellow fever for life. The remaining targets, the ones that are so much more interesting to us as academics and as pediatricians, are much harder because we don't even know if immunity to them can ever exist. So for example, Staph aureus, a disease that I'm sure you all manage constantly in your clinic, it's not clear that any of us are ever going to be immune to Staph aureus, no matter how young or old we are. Whereas for other diseases, an example would be pneumococcus, you are relatively immune after the age of 5 and up to the age of 60, for example. So there's a period of immunity, and then the disease comes back. So depending on whether the disease confers long-lasting immunity, you have an easy or a hard path towards vaccines. And really, I'm making it sound as if this was a choice made by clinicians and vaccinologists in the past, but really what they were doing was a little bit shown on this slide, which is they're doing all sorts of things, and then a miracle occurs and you have protection. And that's the type of process, very empiric process, that led to vaccine development. Now, let me give you another example, which is rabies vaccine. So this is a story that comes from my homeland of France, so I want to tell it to you in a little bit of detail. This is the famous Joseph Meister, who was badly bitten 14 times in 1885 by a rabid dog in Alsace. This was essentially in the 19th century a death sentence. They knew the dog was rabid because he was acting rabid, and the child essentially looked like he was going to die because he had so many inoculations that any clinician at the time would have known that this child was going to die. The dog owner, who was obviously a little concerned, and Meister's father, brought the child to Louis Pasteur. Now, interestingly, Louis Pasteur, although famous and a genius in his own right, was not a clinician and, in fact, not at all licensed to practice medicine. So what he did to this child could have landed him in jail, certainly if it had failed. The treatment was to try to figure out how to prevent rabies in Mr. Meister, in little Meister, and obviously it saved his life. He lived till the age of 64 when he committed suicide upon the invasion of his country by the Germans. So he led a very long life thanks to what Pasteur did. But what did Pasteur do? So what he did is he, basically justified what I said. This is an inevitable death. So even though he was a little bit anxious, he decided to inoculate Meister under the fold of skin with a half syringe full of the spinal cord of a rabbit died of rabies that had been preserved 15 days in a flask of dry air. They made multiple fresh inoculations. I think he did 13 or something. And then on the last day, just to prove that he was right, he injected Meister with live rabies. I don't think this would pass the FDA. I don't know. I don't know what you think. Or an IRB. But this is how the very first rabies vaccine was developed. Now, as you can imagine, and there are well-known cases where the drying in air didn't result in killing off the viral inoculum, and so they injected rabies into patients who died. But the bottom line is this is a very empirical way of developing vaccines. That's the way most of the original vaccines were developed. It can lead to problems, and these are some of the catastrophic complications of vaccine development. As I mentioned, early rabies vaccine that was not sufficiently attenuated, ending up killing the host. There was a catastrophic Qatar incident in 1955 where the poliovirus vaccine that was made, the oral polio, was not adequately inactivated, and so they ended up, unfortunately, killing some patients. And then finally, the formalin inactivated RSV vaccine, something that really was the first, my first foray into the vaccine field, not because I made that vaccine, but it was one that had been given in the 1960s by Chanik and colleagues, and unfortunately they used the same chemical that we use to treat a lot of other pathogens to try to inactivate them and inject them, but in this case it led to immune-mediated exacerbation, and all the kids who got the RSV, or statistically significantly larger number of the kids who got that vaccine were subsequently hospitalized for RSV disease, and in fact 2 of them died. The reason I got involved is that was the actual first vaccine I ever made many, many years later, just as a, shall we say, positive control for disease exacerbation in an animal model. And I hope my career won't be sort of following in those footsteps of the RSV vaccine. There are others that are more just problematic, less catastrophic but problematic. The swine flu, where Guillain-Barré syndrome was associated with the inoculation of a vaccine against influenza. The intussusception risk that you've heard of, a small increase in intussusception risk, which unfortunately led to deciding, probably appropriately in the US, that this vaccine should no longer be administered, even though it could potentially have been life-saving in the developing world at that time. Lyme disease, you know the controversies. I think you had a nice talk this morning about that. There's a recent development of the RTSS vaccine where there's a controversy whether this vaccine actually might increase the risk of cerebral malaria in older infants. So you sort of have transient protection and then maybe increased risk. That topic is still very controversial. Another one that's controversial is the dengue vaccine controversy, where an issue of exacerbated disease can occur when you get a dose of dengue vaccine and then you get another form of dengue that infects you. You are potentially at risk for exacerbated disease, and they're beginning to see that with the Sanofi vaccine that was otherwise licensed in Asia. And then a big problematic issue for all of us is that you can find pretty much anything on the internet about any vaccine you're administering. I played the game once with one of my kids where I said, punch in any disease and any vaccine in Google, and you will find articles describing that association. Hair loss and pneumococcal vaccine. I looked into that for unknown reasons. And then you can look at other things that really clearly indicate that there's a lot of nonsense out there that unfortunately our parents are reading. So if we turn to pneumococcus, the topic of the talk today, the fact is that a mechanism of protection has been known since the early 20th century. So that's great. We know how to prevent this disease. We can make vaccines. And the way we know is that it's very easy to simply say, if you have antibodies to the polysaccharide capsule, shown by these M&Ms here, so the sugar coat of the bacterium, underneath you have the core of pneumococcus, but on the outside you have the polysaccharide, which defines the serotype of the organism. And if you can make antibodies to any of these colors, you will no longer be at risk for an infection by a bug that carries that particular polysaccharide. You will not be protected against a different colored M&M, but you will be protected against the one of the right color. And that's how the vaccines have been made. As you know, Pneumovax, the pure polysaccharide, has 23 essentially colors of M&Ms in the vaccine, or different polysaccharides, and you inject them into adults. They generate antibodies to each of the 23, and they are at less risk of infection by any of those pneumococci that carry that particular serotype. The problem, as you all know, is that children don't respond to pure polysaccharide until at least the age of 2, and probably they don't mature until the age of 5. And then the second issue, which is very important, is that although the Mars company only makes a few colors of M&Ms, there's 97 different serotypes of pneumococcus. And as we knock any one of them down, others pop up, sort of like a whack-a-mole game that you play. You knock down 6B, and then 6A popped up. You knock out 6A, 6C pops up. It's essentially a Darwinian struggle that we are unlikely to win. And so these two problems are the reason why I became interested in pneumococcal vaccine globally, because it seemed to me that there should be a simpler way to try to solve this problem. This is just to show you a micrograph of pneumococcus. This whole thickened layer here is the polysaccharide capsule. And then, as I said, antibodies to the polysaccharide capsule essentially provide protection against that serotype. This is true for other organisms, epiglottitis for Haemophilus influenzae, meningococcal disease, even typhoid. There's typhoid conjugates that are being made right now in Asia to try to prevent typhoid fever. This is a meningococcal disease, and this is periorbital cellulitis from Haemophilus influenzae type B. Adults are different from children, as you know, and so they can respond very well to pure polysaccharides that don't require T cell help. So you basically inject pure polysaccharide, the B cell recognizes the epitope, produces more antibody, and then you get antibody that are non-boostable, but you get antibody to that particular polysaccharide. Repeat doses might be problematic, but 1 or 2 doses are probably fine, and it doesn't have any memory. So every time you redose the patient, it's like the first time they ever saw it, essentially. In contrast, if you actually want to immunize children or get a better memory response, you couple your polysaccharides, shown here in this gray little chains, to a protein. And what it does is it tricks the child into responding to the polysaccharide as if it were a protein. And it elicits T cell help, shown by the CD4 T cells shown here. It leads to many interesting immunological phenomena. One of them is the generation of memory B cells that are ready and primed when you 10 years later get exposed to the bug. Those memory B cells get activated. And the other thing that happens through this process is that the type of antibody you make is better because it matures upon re-exposure to the antigen. So those two factors here explain why a T-cell dependent or a conjugate-like vaccine is probably better than a polysaccharide-based vaccine. So what happened in the year 2000? Several things. Some of you were around and well for this. The inventor, the creator of Peanuts died. That was very sad. Some of you remember Napster. That was when there was the first lawsuit against Napster, which resulted in the destruction of that company. There were some other events here, Bush v. Gore, all of these things. But the most important one from an immunological perspective is that was the year that Prevnar 7, which contains 7 serotypes of pneumococcus, was licensed in based on studies in Northern California. We were all very thrilled about that. As shown on this slide, people thought this is finally, we're going to attack and destroy all pneumococci because those are the 7 most common ones. and we don't know what whack-a-mole means. I don't even know what the game is. This thing is going to just eliminate 89% of all pneumococcal disease in the US and worldwide. Since then, people realize that's not quite the case. You have to expand because other serotypes emerge. So GSK made a PCV10 vaccine that is not licensed in the US, mainly not licensed because it is not proven to be non-inferior to Prevnar. So they were unable to show that their titers were as good as Prevnar, and so they didn't even bother to go into the US system, but they are licensed in Europe and other countries in the world. And then, as you know, in 2010, Prevnar 13, the one you're using now, was licensed, which included 6 additional serotypes. The most important one was 19A, which was really causing a lot of disease after PCV7 was introduced. And in fact, a separate conversation, there's good evidence that PCV7 led to the emergence of 19A through that process. Normal process, nobody's fault, but 19A was there, lurking, and then it took advantage of the fact that those 7 serotypes were no longer present. We know today that Merck is working on a PCV15, which is essentially PCV13 plus 22F and 33F as 2 serotypes. And then we've heard rumors, and there was a press release from Pfizer, that they're working on another one, but they didn't say how many serotypes it would cover. So we don't know. The overall impact, again, is pretty obvious. There's a tremendous reduction of the 7 and then 13 serotypes that cause disease. A simple fact is that if you see tomorrow, by some unfortunate luck, a child with invasive pneumococcal disease, the likelihood that that child has the 13 serotypes, one of the 13, is vanishingly small. They're almost certainly going to be infected with a non-vaccine serotype. So that's a phenomenal impact of this vaccine. It has a huge impact of herd protection that I'm going to show you in a few seconds. So vaccinating infants protects adults. That's great. There's some clear efficacy against mucosal disease like pneumonia. It's harder to measure because in most cases of pneumonia, we don't know what serotype infected the child, or even if it's pneumococcus. Another problem is that these vaccines are very expensive and hard to make, and that was partly why I got into this field as well, is to try to make something that might be a little bit less expensive. And then finally, the issue of serotype replacement. So the cartoon, for those who can't read, these are 2 bacteria in a flask. I'm fed up with this guy. Let's become pathogenic. It's the idea that the serotype replacement results in strains that become more and more fit to then, unfortunately, as a consequence of fitness in colonizing the mucosa, are able to infect individuals and make them sick. That's not their purpose, obviously. Their purpose is just to colonize, presumably. But in their attempt, in these bacterials' attempt to really be better at colonizing, you have that unfortunate side effect. So one of the lessons today is that the only thing that matters, not only in pneumococcal disease, but probably for the best vaccines we have, is blocking transmission. The best vaccines you all use, Haemophilus influenzae, meningococcal vaccine, pneumococcal vaccine, measles, the old whole cell pertussis, not the new one. All these vaccines were phenomenal, or are phenomenal, because they block transmission. So if you vaccinate one kid in daycare, he is not going to infect or colonize other kids in daycare. So take Louis Pasteur. He discovered pneumococcus in 1881, and he has pneumococcus up his nose, for example. He then meets our boss at Children's, Gary Fleischer. But Gary Fleischer, who some of you probably know, is a very strong, powerful man, is impervious to pneumococcal carriage. He cannot be colonized. We don't know why. Actually, some people are less likely to be colonized than others. That's something my lab is interested and has been studying for years. But so Gary will not be infected by Louis no matter what happens. However, other people might. So this is my mentor, Porter Anderson. This is Louis Sternberg, who was the co-discoverer of pneumococcus, although he doesn't get as much credit as he deserves. And then this, of course, is Mark Waltzman. All these people can get colonized by pneumococcus through one index case, Louis Pasteur. But none of them will get sick. They will just hang out and be perfectly happy and serve as little bioterrorists in the community. And what might happen is this, is Mark worked with me a few years ago in the travel clinic. This is what I looked like. I'm not kidding. It's true. And this is a true picture. It's not photoshopped, I promise. But this is me. And he colonizes me by just not normal interaction. We were very close. And so he colonizes my nose. And as a consequence, for reasons that we don't quite understand, sorry if you can't see, if I'm in the way, for reasons we don't totally understand, there's the possibility that I develop invasive disease, We don't know why I get invasive disease and Porter doesn't, and why all these others are not at risk. And it might be genetic, it might be exposure, we don't quite know. But the bottom line is that this can be prevented if you block every other stage of the process. And that's what Prevnar really is doing. We know that Prevnar is preventing disease in Louis Pasteur if he gets it, if he gets the vaccine. But what it is really doing is it's preventing Louis Pasteur from spreading the organism in the community. And that's probably the single most important thing that these vaccines do. Why do I say this? Because when you vaccinate children in the US, and this is data from 2005, so between the Prevnar 7 and after 7 and before 13, so you got about per year 9,000 cases of pneumococcal disease prevented, a phenomenal impact of any vaccine, no question. But even better yet, the elderly, you saw a reduction in 20,000 cases. So twice as many. And they were not being vaccinated. So now they are, but then they were not. So the only possible explanation for this is indirect protection by blocking transmission. And that's why Prevnar is such a phenomenal vaccine. This is a paper in the New England Journal that just shows the same thing, that you are preventing pneumococcal pneumonia and invasive disease thanks to this vaccine, a tremendous impact. So one of the most important things to tell you is that this is what we're trying to prevent. We are trying to block transmission. And in fact, although it's true that you can prevent invasive disease by neutralizing the bacterium in the bloodstream, by generating anti-capsular antibody, the key reason why all these vaccines are so successful, GSK's, Prevnar, all these vaccines are so successful is because they're blocking transmission to susceptible hosts. So high concentrations of these antibodies are probably preventing colonization in the nose, and probably doing it by agglutinating them and allowing the immune system to get rid of them as fast as possible. So, why should I be spending my time working on pneumococcal vaccines? One of them says, oh, I found a cure for pneumonia, and the other scientist says, that's going to cut off our funding. Of course you haven't found a cure for pneumonia. We have to hide these data. So why would anybody worry given this? And as you know, one of the reasons is this tremendous cost of this vaccine. It's over $100 per vaccine if you buy them, without insurance, for example. This is Fan Zhang, a very talented scientist in my lab, who I'll talk to you about in a few minutes. But we had to do a big animal experiment, so she was carrying thousands of dollars in her hands of Prevnar, because we had to buy it, of course, not for patient use, so we paid, full cost for these vaccines, which is why I know exactly how much it costs for Prevnar 13. Another reason why we have to work on better pneumococcal vaccines came from another friend of mine, Mark Lipsitch, a mathematical modeler at the School of Public Health. And this is a paper that was published in 1997 in the Proceedings of the National Academy of Science in PNAS. This was way ahead of its time because Prevnar had not even been licensed at that point. But he predicted, and all of you in the back of the room, I'm sure, understand what these symbols show. I still don't understand him, even though I've known him for 20 years. But he predicted, using mathematical modeling, that we would absolutely see serotype replacement. When he wrote the paper, it got published, and everybody, even my mentor, another, not Porter Anderson, but somebody else, said, that's nonsense. We're just going to wipe out pneumococcal disease. And in fact, that's not at all what happened. In the UK, this is the lay of the land before they introduced Prevnar. So you see predominant serotypes shown by these pies in the nose of children. And then after the introduction of Prevnar, all these new different colors and new numbers that we had never heard of before emerged. Serotypes that I had never even considered could cause disease suddenly emerged in the UK and were important causes of disease, as shown here, like all these different serotypes. So again, it proved that by targeting 7, you were going to end up with more, and by targeting 13, we have now ended up with other ones as well. The total number of cases is dropping, that's great, but you're not eliminating the problem altogether. In the UK, they actually found that herd immunity in the elderly was in fact shown here, was compensated, unfortunately, perfectly by a rise in non-vaccine types. So this is age over 65 years, and the green line shows the drop that we saw in invasive types that infected these elderly in the vaccine, but you have an absolute equivalent rise in the red line, which is the non-vaccine types, so that the blue line, which represents all pneumococcal disease in the 65-year-olds, stayed the same. different from the US. In the US, we saw this nice drop. And it's just because the serotype distribution in the UK and the US is different, we presume. But also suggests that the vaccine is not the panacea we hoped it would be. We published on this, if anybody's interested, in 2011. A graduate student at the time, Dan Weinberger, Mark, and myself published a paper to try to explain the impact of serotype replacement. But importantly, and this is maybe another important message today, this is not what's happening. We're not sort of taking, a burning building patient and bouncing him into another one. That's not at all what's happening. We're clearly impacting pneumococcal disease. Some of us just want to be able to do better. So, how would you do better? One sort of premise that we had, or two premises that we had, was that given the success and the nature of the conjugates, the fact that they prevent disease in the individual, and the fact that they prevent transmission, any vaccine we propose to develop has to do those two things. Both provide protection against invasive disease and against carriage. Without one or the other, the vaccine is a non-starter in our minds. So that was our premise. You have to do both. So, what's the solution to try to improve? The most obvious solution is put more capsular serotypes in the vaccine. So Pfizer made 7, then they made 9 actually, but never got licensed. Then they made a 13-valent. 13 is better than 7. And Pfizer announced that they're going to expand Prevnar 13, but they didn't indicate the valency. And the question is, technically, how high do you go? We don't really know. Merck is making a PCV 15, which won't really move the needle that much, but it's an interesting addition. And then there are other vaccine manufacturers that are working on trying to make a broader valency, but they haven't released the number of serotypes that they want to include. And as now a vaccine developer, I can tell you that there are clear manufacturing hurdles when you take 13 serotypes and want to then increase it even further. Because as some of you probably know, making a 13-valent vaccine is making 13 vaccines. It's not making one. And making 13 vaccines in the current climate, which is a very good one at the FDA, means thousands and thousands of QC tests that have to be validated. And it's, $800 million or more to develop any one of these vaccines, plus all the technical problems. And then from my very biased perspective of somebody interested in developing world, these are procedures that developing world manufacturers probably can't follow with the same degree of technical advance that we can in the US. I mean, no disrespect to developing world manufacturers, it's just they don't have the same technology, they don't have the same resources that we do. They certainly have developed fantastic vaccines and are proceeding in that direction. But to make Prevnar 13, it is the single most complicated vaccine ever made in the world. I'll give you an example. I spent many years working in Brazil on one of the projects I'm going to tell you. And Brazil wanted to introduce Haemophilus influenzae conjugate type B vaccine. One vaccine, one conjugate. So they established a contract with GSK, where GSK would teach them how to make it, and at the same time, until they were ready, would sell them Hib, the vaccine. That was 20 years ago. They're still buying Hib from GSK. They have still not been able to manufacture their own conjugate because of how complicated it is. So, and that's Brazil, a very well-developed country. So these things are just, they take a lot of time, they take a lot of resources that unfortunately are only available in the most wealthy countries in the world. And then another problem with this approach is that the whack-a-mole process might just continue. You target 20, and then another 20 pop up, and ultimately, at some point you hope that you've just targeted the most important ones. And I think we're approaching that with Prevnar 13. We're coming close, and maybe Prevnar 15, Merck's 15 will do even better. But that's sort of the balance that we just don't know what that magic number will be. So one option that we developed at Children's, this is not in a company, it's developed by the Gates Foundation currently in PATH, is to make a whole-cell pneumococcal vaccine just like pertussis. So current vaccines, as you know, are based on polysaccharides, and we decided in 1996 to revisit this issue and basically take the whole organism, strip the capsule away genetically, get rid of it, and mutate one of the toxins so that it doesn't hurt the recipient, grow it up to high concentration, kill it, purify it, inject it into people, and see if they're protected by virtue of all the proteins that constitute pneumococcus. So another you'll forgive me, I hope, for the simplicity, but immunizing with the chocolate of the M&M. not the polysaccharide capsule. And that was our plan with Porter Anderson. It was his idea in 1996, and I quickly enlisted on the project. In fact, this had been done by this man, Sir Almroth Wright, in 1911 in South Africa. He did a slightly different, wasn't quite the same procedure, but he was the father of the whole cell typhoid. He said, I'm going to do the same thing for pneumo. He was called by South Africa, which in those days was very close to Britain in terms of their politics and their rule. And so he went down to South Africa and he said, I'm going to help because there's an outbreak of pneumococcal disease in minors. And we're going to take the prevailing strains, grow them up, kill them, and inject them. So that's what he did. And he vaccinated 50,000 minors with a killed prep of his pneumococcus, and he claimed success. It worked. The epidemic went away. Now, unfortunately, others contested his findings, nicknamed him Sir Almost Right instead of Sir Almost Right, and they basically said, his statistics were flawed because he didn't have a control group. So, of course, how can you prove that the disease went away and by your vaccine and not, in fact, by some other process? So people kept on testing this, though, because it was so elegant in terms of its simplicity. And so the military tried it quite a bit in the 1940s. And if you're interested, there are papers that show more often than not that it did reduce disease, even when it was well controlled. So, but we couldn't say exactly by how much. The studies were very small in number. There were some in the military that were larger, but enough to make us interested in doing it again. So I won't go over all this, but my lab, with Porter Anderson as my mentor, has basically taken the same approach, and we were able to show that you could basically immunize with these bacteria, these killed bacteria, through different routes, and you show a two-pronged mechanism of protection. One of them is antibody-mediated against sepsis, and the other one is T-cell mediated, CD4 T-cell mediated against carriage. So the two premises that I was telling you about, prevent carriage and prevent disease, it appears that this vaccine in mice and rabbits was able to do. So we were happy with that. But of course, as the slide shows, mice like to trick humans. And so these mice are saying, when Porter and Rick come in, everybody play dead. And that's in fact what we were worried about, that there may be no evidence for this type of mechanism in humans. So we focused on the T-cell one because that was kind of novel and interesting. And it turns out that there's enough circumstantial evidence that humans are protected, not by vaccine, but by natural exposure with pneumo, probably through T-cells. So we all think of antibodies to pneumococcus as being critical, and that's true. They probably prevent disease. They certainly can block carriage if you have enough of them against the capsule. But what's interesting is that if you take HIV patients, for example, they have a ton of antibodies by the time they get HIV. They're normal, healthy adults until, unfortunately, they get HIV. And yet their risk of pneumococcal disease goes through the roof, unless they're treated with heart and so forth. So this suggests that T cells are very important in control. And in fact, when you go to Africa, the HIV-positive patients are loaded with pneumococcus in their nose. So at least there's the suggestion that you have a T-cell deficiency, and in association with that, you have a high rate of carriage. We also know that individuals who have low CD4 T cells, reactivity to pneumococcal proteins, are at higher risk of being carriers. We've demonstrated that as kids age, they develop better Th17 responses, which at least might correlate with a resistance to colonization. And then most importantly and recently, the human knockout of Th17 are Job syndrome patients. These are individuals who have a STAT3 mutation, and it's very rare actually. I think we all learned about it in medical school, but it's quite rare where these individuals have boils due to staph, recurrent pneumonia due to pneumococcus and staph, they have facial deformities, all sorts of other things. But their primary defect immunologically is that they can't make Th17. And what diseases are they susceptible to? Pneumococcus, staph, and Candida. Those are the three. And in the lab, you can show that pneumococcus, staph, and Candida are, in mice, you can block those diseases through Th17 mechanisms. So again, circumstantial evidence that suggests maybe we're not completely off. And then finally, a more recent paper showed that if you look at children who have slight mutations in the IL-17 gene, they're more at risk for carriage with pneumococcus. So enough data to make you think, if you could harness this and jumpstart a 1-month-old into developing that response by 2 months instead of by 5 years, maybe you can drop his risk of being a carrier and protect him and also the community. So where did the whole cell vaccine go? We did a phase 1 clinical trial in healthy adults in Seattle. Vaccine looked really good. We then moved to Kenya, where we are now immunizing adults and toddlers. It's ongoing, but so far so good. No safety signal that we can detect. And we're currently in plans of a phase 3 clinical trial in Indonesia, a country that cannot afford Prevnar. So they're very interested in an alternative, where the vaccine is actually being made. And then just as a brief word, we're not interested in providing a country with a lesser version of Prevnar. I mean, my vision is not to use the developing world as a testing ground for vaccines that are inferior to what those children deserve to receive. That said, Indonesia cannot afford Prevnar, and until the vaccine companies are willing to sell them at a much lower price than they are currently, we have to try to find an alternative. And maybe the whole cell vaccine would be as good, maybe not as good, or maybe even better, because it would potentially cover all strains of pneumococcus, not just those that are covered by Prevnar 13. Because again, this is an unencapsulated strain, and just like pertussis is generating immunity to the old cell pertussis is generating immunity to a number of proteins on the surface. The advantage is it's very simple to manufacture. I could teach any of you to make it in your kitchen if you want. It exposes the host to numerous antigens. There's some suggestion, as I mentioned, of efficacy historically, and then you have all these proteins that you're injecting with. Incidentally, I tested it on myself and Porter on himself before we went to phase 1 clinical trials, and except for the way I look, no obvious side effects were noticed in us, and that was one way to allow us to then proceed with a reasonable idea of what would be a tolerated dose. Disadvantages, it's a complex antigen, and I can't tell you what is protecting the individual. So going back to the first slide of how do we make vaccines, this is grossly empiric. We're just blasting them with the whole organism. So it's very hard to define the mechanism of protection. How are you going to license this? Is it going to be acceptable in the developing world? And then if it works really well, could it ever even potentially work in a more developed setting? We don't know. I think it's unlikely today that some in the developed world would accept a vaccine like this unless its impact is truly phenomenal. So, what if this doesn't work? And that's either probable or possible, depending on whether you're a pessimist or not. And there's lots of reasons that I won't go over why it may not work, but one of them might be it just may not work. So what if it doesn't work? So again, we would want a vaccine that is low cost. That to me is one of the most important things, because otherwise we might as well stick with Prevnar. You want a vaccine that elicits antibodies, and ideally, you would like to try to generate protection against all pneumococci, not just the ones that are covered by Prevnar. So, to remind you, conjugate technology is based on coupling a sugar coat, this sugar strand, to a protein shown in red. And we do it, the protein could be CRIM, as in Prevnar, it could be tetanus toxoid, the outer membrane protein complex of meninge, which has been used for Haemophilus influenzae, protein D, which is the protein they use in Synflorix, for example, the PCV10 made by GSK that we don't use in the US. So all of these proteins here don't really contribute to protection against the organism. They're important carriers, and they trick the body, the baby, into responding to the sugar as if it were a protein. But they are not really contributing anything else. So could they? Could you put a pneumococcal protein here and then sort of have that two-pronged, serve as a carrier but also as an antigen? So we developed a technology, very simple technology at Children's, where you put tags on proteins, so-called biotin binding tags, and then you put biotin on your sugar. And biotin binding tags and biotin find each other and stick like Velcro. So for any of you who's ever spent one day in the lab and done an ELISA, that's how ELISAs work. You tag an antigen with an antibody, and then you go after that antibody with a biotin-avidin or strep-avidin interaction. So we use something called risavidin, but it's a very similar thing. And we can take any pneumococcal, TB, staph, any protein we want, and tag it with this biotin-binding protein. You stick them together, and you get a molecule that looks like a conjugate, but is not a conjugate. It is, in fact, very different on electron microscopy. Instead of looking like a big meatball and spaghetti like most of our vaccines do, because they're strands of sugar, polysaccharides with proteins, and they're all cross-linked and so forth, in our case it's like beads on a string. So you have the string of polysaccharide and then these fat beads of proteins that attach at a single point. And that's what we call MAPS, the multiple antigen presenting system. It has some very nice properties. It's very easy to make because you don't have to be a chemist. I'm not a chemist. You just have to mix two things together and they stick together. It's very modular, plug and play. We can make these things for any number of organisms. And then we found when given with aluminum phosphate or hydroxide, the typical adjuvant we use in Prevnar, that you can get not only antibody to the polysaccharide, you get antibody to the protein, and interestingly for my lab, you also get Th17 responses to these proteins. The same thing we were hoping for in the whole cell vaccine. People ask us, could it be dangerous to be injecting biotin and rhizavidin into people? The answer is we don't know yet because we haven't done it, but in rabbits and mice and in guinea pigs, they seem to tolerate it just fine. But of course, the test will be in phase 1 clinical trials in adults and subsequently. And we have not noticed any cross-reactivity between rhizavidin and avidin. Somebody also asked, well, could you generate egg allergy if the rhizavidin looks like egg avidin? And interestingly, people who are allergic to eggs generate antibodies IgE class, but not to avidin, to other proteins of eggs. So the good news is it doesn't appear like it's not like we picked ovalbumin, which is one of the agents of allergy in egg allergic patients. So this is a slide just to show in red the maps that we made. It's a MAPS 13, and we compared it to Prevnar 13. This is one dose in rabbits, and with one exception, which I will show here, the 7F, in every other case we found that our vaccine actually outperformed Prevnar in rabbits. Now, this is rabbits, it's not infants. Prevnar was optimized for infants. We haven't been in infants yet, but it was very promising that we were able to make a Prevnar 13-like vaccine and generate the same immune response, if not better, in rabbits. The T-cell response also enables us to protect mice against carriage, which was another plus. And so we were very encouraged by this, that we can actually make something like a whole cell vaccine, but without the complications of a whole cell vaccine. It's generating good anti-capsular immunity, and it's also generating protection against carriage. So, in summary, MAPS, the technology we've developed, is one approach of many that you could use. The whole cell vaccine, something that Children's also developed, is another approach, maybe more targeting for the developing world. The experience now in our mind with conjugate vaccines suggests that what's most important is to try to not only protect the individual, but also confer indirect protection by generating a blockage in transmission between children and prevention of transmission from a child to an elderly patient. We think that there's a very good reason to consider alternative strategies, whether it's the whole cell, MAPS, or anything else, because of the tremendous cost of these vaccines for the developing world. And I think you can try to adapt any of the things, any of the concepts that I've mentioned today, and try to come up with a better strategy, some other companies are, than what we're doing at Children's, to try to come up with the ultimate pneumococcal vaccine. And so these are all the people that have done the work or have provided funding. Yingji Liu and Fan Zhang are married. They were married before they met me, but they're a married couple in my lab. Fan is the young lady who is carrying the Prevnar. Yingji is, the three of us are co-founders of the company that I mentioned, scientific founders. And then my boss at Children's, Michael Wessels, who has provided a lot of support over the years. Thank you very much for your attention, and I'm happy to take any questions.